Good afternoon, everyone. I'd first of all like to start by thanking the meeting organizers for the opportunity to come and speak on behalf of Argos Therapeutics. Argos Therapeutics is a late-stage biotechnology company focused on developing individualized immunotherapies for the treatment of cancer and infectious disease. For the purpose of today's discussion, I'll focus on our lead asset, AGS-003, which is being developed for the treatment of cancer. We'll be making forward-looking statements throughout the presentation. So what differentiates the Argos technology in the complex and competitive field of cancer immunotherapy? The first key attribute is our novel platform technology called Arcellus. The novel platform technology utilizes a total tumor messenger RNA approach where we load autologous dendritic cells and leverage tumor neoantigens to stimulate an immune response that's been shown to correlate with patient survival. The second key attribute is having an advanced clinical program. We recently, in July of this year, completed enrollment to a phase 3 registrational trial in frontline metastatic kidney cancer that's under SPA with the US FDA. We expect interim data from the study in 2016 and final data in mid-2017. The third key attribute is having broad applicability. Given the fact that we take a total tumor messenger RNA approach, any tumor type where we can get a small tumor sample and we've produced this product from as little as 5 milligrams of tumor tissue is a potentially viable target for future clinical development planning of AGS-003. The other key attribute in the direction that the field of cancer immunotherapy is moving is all about combinations. So one of the other key things that will be important for our development plans as we move forward, given the fact that we've tested AGS-003 now in over 300 patients to date and have yet to see a serious adverse event attributed to the product, we will look at developing this therapy in combination with standard of care, as well as with other immune checkpoint inhibitors targeting PD-1 and PD-L1. The fourth key attribute that differentiates Argos is having an optimized commercial model. One manufacturing run for us with one leukapheresis sample allows us to produce on average 3 to 5 years worth of dosing for a patient. This is currently done under a manual manufacturing process, and as we transition over to a commercial stage company, we will utilize our partnerships with companies like Invitech as well as with Saint-Gobain to implement automation that will further reduce our cost of goods as well as increase scalability. So how does the product work? The product works by activating naive CD8 positive T cells and differentiating them into CD8, CD28 positive memory T cells that have been shown to correlate with good clinical outcomes. In order to stimulate this T cell activation, we leverage two key components to the product. The first component, given that we take a total tumor messenger RNA approach, is leveraging the neoantigens to stimulate an immune response. The second key component that we leverage is the addition of CD40 ligand messenger RNA, which is the only non-autologous component that is added into the product. This allows us to overcome tumor-induced immune suppression that comes from regulatory T cells and myeloid-derived suppressor cells. So by adding in the synthetic CD40 ligand RNA, we reprogram the immune system and allow it to function as if it was an immune-competent host. This next slide provides further detail around the manufacturing process. As you can see here, there are 3 key components in order to produce the AGS-003 product. The first of which is a tumor collection. From the tumor collection, we isolate messenger RNA, which is specific to the antigens present on that patient's tumor surface, and we amplify them using RT-PCR, which is why we need such a small tumor sample. The second key component is the leukapheresis procedure or cell collection. From that leukapheresis sample, we isolate the monocytes and maturate them via a process of proprietary sequential cytokine treatment into fully mature dendritic cells. The final key component that goes into our product is a synthetic CD40 ligand messenger RNA So the final AGS-003 product is the maturated dendritic cells electroporated or loaded with the total tumor messenger RNA and the synthetic CD40 ligand RNA. This is all done currently under a manual manufacturing process. One manufacturing run through this process allows us to produce 3 to 5 years worth of dosing for the patient. Each of those doses are aliquoted out and cryopreserved and delivered on an as-needed basis. The product is administered by an intradermal injection, most commonly in the axillary lymph node bed, where it migrates into the immune system and exerts its effect. The dosing schedule for this product comes in 2 phases, the induction course and a booster phase. During the induction course, the product is administered as 1 dose every 3 weeks for a total of 5 doses. Patients then go on to receive quarterly booster injections. So now that we've talked a little bit about the science and the manufacturing, let's talk a little bit about the clinical data. So prior to moving into phase 3, Argos completed a phase 2 combination study looking at AGS-003 with sunitinib in newly diagnosed patients with metastatic renal cell carcinoma or kidney cancer. Prior to diving into the clinical data, it's important to have some background on metastatic renal cell carcinoma. Patients with metastatic renal cell carcinoma are split into 3 risk classifications: favorable, intermediate, and poor risk. Each of those risk classifications has a varying degree of survival. Patients with favorable risk disease have an expected median overall survival of about 42 months. Patients with intermediate risk disease have a median overall survival expectation of around 22 months. And patients with poor risk disease have an expected median overall survival of about 8 months. Argos has decided to focus its initial development on the unmet medical need that exists in this patient population, which is patients with intermediate and poor risk disease. As you can see from this slide, the phase 2 combination study was a single-arm study, so the comparison that you see here on the slide is to historical control. The phase 2 combination study enrolled a patient population that was 48% poor risk and 52% intermediate risk, producing a median overall survival of 30 months. This is double the expected median overall survival for current standard of care, and as you can see in the historical control arm, patients treated with frontline Sutent or tyrosine kinase inhibitors, even though that patient population had a better prognosis, have a median overall survival expectation of 15 months. And this comes from a historical control of 1,200 patients recently published last year by Danny Hang and colleagues from the International Metastatic Renal Cell Consortium. In addition to the doubling of median overall survival, we also saw a doubling of the progression-free survival and a 43% partial response rate. To further take a look into the phase 2 combination study, this next slide shows a waterfall plot of all 21 patients that were treated on the study. I would like to draw your attention to 2 key aspects on this slide. The first of which is long-term survivors in metastatic kidney cancer. A long-term survivor in metastatic kidney cancer is defined as a patient who survives for greater than 30 months. The expectation with frontline Sutent is 13%. We saw more than a 4 times improvement in this, and that 52% of our patients on the phase 2 combination study survived for at least 30 months. The other key thing on this slide that I'll draw your attention to are patients 20 and 21 on the waterfall plot. You'll see that both of these patients are still ongoing AGS-003 booster dosing and are now out over 6 years on treatment. Both of these subjects remain progression-free and in long-term remission. Sorry for the delay with the slides. In addition to the clinical benefit that we saw in the phase 2 combination study, we also saw a biological signal in this study through the translational research that was part of the phase 2 combination study. On the phase 2 combination study, we utilized 17-color flow cytometry to look specifically at the CD8, CD28 positive T cells, so the specific T cells that we actually generate with our product. We looked at these at baseline, after 3 doses of AGS-003, and after 5 doses of AGS-003, and we looked at their correlation with relevant clinical outcomes, including survival, PFS, and tumor response. If you look at each of these time points individually, so baseline, after 3 doses, and after 5 doses, there's no correlation between those time points and any of the relevant clinical outcomes. If you look at the difference between baseline and after 3 doses of AGS-003, so truly the CD28 positive T cells that are stimulated with our product, you start to see a trend towards a correlation with those relevant clinical outcomes. After 5 doses of AGS-003, so the difference between baseline and after 5 doses, there is a strong statistically significant correlation between the magnitude of the immune response generated by our product and the duration of survival to a p-value of 0.002. In addition to the correlation with survival, we also saw the same correlation with progression-free survival and also with tumor response. So as we transitioned from the phase 2 to the phase 3 study, there were really 2 key enhancements that we made to the study design. The first of which is dosing through progression. On the phase 2 combination study, the traditional RECIST criteria was applied, and as soon as a patient had an initial progression, they came off study. When we discussed the immune monitoring data from the previous slide I just showed you with the US FDA as part of our SPA negotiations, the first question they asked us was, why are you not dosing through an early progression to give patients the best shot to get at least 5 doses of AGS-003 on board? So that's exactly what we're doing on the phase 3 study. The second key enhancement that we made was that there was a much larger benefit in patients with intermediate risk than in poor risk. So rather than just eliminating patients with poor risk disease, we focused on patients with 1 to 4 risk factors. And the phase 3 study enrolled 73% patients that were intermediate risk and only 27% that were poor risk. So a much higher percentage of intermediate risk patients enrolled on the phase 3 study compared to the phase 2 combination study. This slide provides an overview of the phase 3 study design. This phase 3 study is now completely enrolled. We enrolled 450 patients. It's a 2-to-1 randomization. Patients either got AGS-003 plus standard therapy versus standard therapy alone. Standard therapy on this study, all patients start on Sutent to leverage the immunomodulatory effects of Sutent. But in the event that a patient has intolerance or if they have progression, just like in a real-life scenario, patients are allowed to switch to another tyrosine kinase inhibitor or mTOR inhibitor. The study is powered to detect and show a 6-month improvement in overall survival. That assumes a little over 16 months for control and a little over 22 months for the combination. We expect data from this study, interim analysis data from the 50 and 75% interim in 2016, and final data in mid-2017. While we wait for the data to mature from the phase 3 study, we're also starting to investigate earlier lines in kidney cancer, as well as investigating other solid tumors. We currently have an ongoing study being done at Roswell Park Cancer Institute in neoadjuvant RCC. And in the next 6 months, we'll be opening studies in adjuvant non-small cell lung cancer, as well as in neoadjuvant bladder cancer. In closing of my talk, I'd like to thank the patients and the investigators that participated and made the success for the ADAPT study enrollment. And I'll take any questions from the podium or after. Thank you.